So, good evening, everyone. My name is Mariam, and I'm the Education Manager of the ERN-EuroBloodNet. It's my great pleasure to welcome you on behalf of the ERN-EuroBloodNet to the third webinar of the topic on focus on constitutional thrombocytopenia. So this series is an accredited European online education program that targets health professionals and that is organized by the ERN-EuroBloodNet in partnership with the CRPP, the French Reference Center for Inherited Platelet Disorders, and the Filière MHEMO, the French Constitutional Bleeding Disorders Network. This whole program consists of a cycle of 15 webinars developed in collaboration with 3 experts, Professor Marie-Christine Alessi and Dr. Saultier, who we have with us tonight, from Assistance Publique-Hôpitaux de Marseille, who are a member of the ERN, and Dr. Mathieu Fiore from CHU Bordeaux. So our goal with this program is to disseminate up-to-date knowledge among interested hematologists, internists, pediatricians, and healthcare providers to give visibility to cutting-edge advances in the field of constitutional thrombocytopenia. Before we start, please note that this session is recorded and will be uploaded in the EuroBloodNet e-learning environment. If you do not wish to appear, so please turn your camera off and change your name on the platform. Please be also kindly informed that your microphone is muted. And if you have any questions about the presentation, please write them in the chat. Try to make sure to mention which specific slides you're referring to or which specific topic you're referring to, and they will then be addressed during the Q&A session at the end of the webinar. You can access the chat by clicking discussion on the bottom of your screen. As mentioned, this program is accredited by continuing medical education points issued by the European Body of Accreditation in Hematology. You can request 1 credit per session or 15 credits if you follow the whole program. So to request credits, you can simply make sure that your name and surnames are visible on the platform, then fill out the attendance survey that will be linked in the chat, and finally answer the 3-question short satisfaction survey that you will receive after the webinar. So today is the 3rd webinar of this new series. Please join me in welcoming Professor Andreas Greinacher, who will lead this lecture entitled Blood Smears Combined with Immunofluorescence Facilitate Diagnosis in Patients Suspected for Inherited Platelet Disorders. So Dr. Andreas Greinacher is specialized in transfusion medicine, immunohematology and hemostasis, and is full professor and head of the Department of Transfusion Medicine and the Thrombosis and Hemostasis Service at the Universität Medizin Greifswald in Germany. His research interests are hereditary and immune-mediated thrombocytopenias, especially heparin-induced thrombocytopenia, and application of biophysics to understand molecular mechanisms of antigenicity of endogenous proteins. Professor Greinacher, the floor is yours.
Okay, yeah. Thank you very much for the kind introduction and welcome everybody. And if you, as participants, don't mind, I wouldn't mind if some of you switch on your cameras. It's much easier for me to give an enthusiastic, lively talk if I see at least a few faces instead of only my blunt computer screen. And today I will introduce you to some work which has been spearheaded in our group primarily by Carlo Sanninetti, and Carlo is also online in the moment with us. And I'll speak about the potential to use the old-fashioned blood smear to facilitate the diagnosis of inherited platelet disorders. These are my conflict of interests. None of them is related to our current topic of today. The learning objectives are that the platelet phenotype can be characterized using blood smears and immunofluorescence and take advantage of the many monoclonal antibodies we have, which can characterize and visualize specific platelet structures. And by this, the typical morphological features are supportive in coming to the diagnosis of a specific inherited platelet disorder, or even if the features are not specific for a certain disorder, sometimes one can conclude even therapeutic measures from the disturbed platelet morphology. Let's say if the cytoskeleton is disturbed, one knows that the platelets are not able to really stabilize the thrombus well enough. And in this situation, fibrinolytics are an important measure to improve hemostasis. You all know that production of platelets is a quite complicated process. First, TPO has to bind the receptor. Then we have a cascade of signaling leading to activation of transcription factors. And then the megakaryocytes develop by endomitosis, undergo cytoplasmatic maturation. Finally, the megakaryocyte then develops proplatelets, and these platelets have to be released, which means in such a complicated process, mutations in many different genes can cause a platelet-related bleeding disorder. You all know that characterization of genetic disorders usually requires a very good phenotype of the patient to make sense of the genetic disorder and to put the genetic disorder into a perspective fitting with the phenotype. The problem, however, is that in many of these platelet disorders and platelet function disorders, the defect is moderate and the bleeding disorder is also moderate. It's typically menorrhagia, mucocutaneous bleeding, bleeding after surgical interventions like tonsillectomy or tooth extraction. But these symptoms are very unspecific and more or less account for all of the platelet function disorders. And we can draw some conclusions from the mode of inheritance, whether it's a dominant or a recessive disorder. But also this is not very indicative for a certain type of platelet disorders. And then the problem for most of the specialized laboratories are that patients often live quite far away from the laboratory and have difficulties traveling. And many of our methods still require relatively large blood samples, which is an issue for pediatric patients and becomes an unsolvable issue, especially in newborns in whom a certain platelet disorder is suspected when it comes down to flow cytometry, platelet aggregometry, Lumi aggregometry, or other methods which require platelet volumes in the range of a couple of milliliters. Now, next generation sequencing gave us a perfect perspective. It can be performed with minimal amounts of blood. The pre-analytical factors can usually be controlled very well. The samples can be shipped by mail to central laboratories, and during the last years, the costs really decreased a lot. And I've seen Anna Savoya being here in the audience, and she is really one of the most experienced scientists in the field, and also the Spanish group with Dr. Rivera, who is also with us today, has lots of experiences with this. Now, what are the potential issues and problems? One of the main problems is how to interpret variants of unknown significance, and quite a large proportion of the identified gene changes are variants of unknown significance. Deletions and copy number variants are sometimes difficult to diagnose. Patients may have several compound variants which only together create a phenotype. And the problem of good phenotype characterization can not be obtained by clinical history and the simple bleeding phenotype, because this is rather unspecific. And this led us a few years ago to team up with some of our colleagues whom you all know very well: Alexandra Peggi, Shinji Kunishima, Patricia Nörden, and Tamam Bakshul from Tübingen. We all teamed up and investigated systematically whether we can use blood smears and immunofluorescence to phenotype platelets by microscopy, because in a platelet disorder, the phenotype of the platelet is leading towards the diagnosis. We have different inherited platelet function disorders. All of you know this. Sometimes structures absolutely required for platelet function are affected, and Glanzmann thrombasthenia and Bernard-Saultier syndrome are a bit like this car, which lacks the tires and the wheels, although the rest of the car is perfectly normal. And the storage pool deficiencies, the dense granule deficiencies, are a bit like a car with an empty tank running out of gas. I start with platelets with normal size platelets and normal numbers, the storage pool deficiencies, Glanzmann thrombasthenia as an example. And aggregometry is often used to diagnose these disorders, but platelet function is complex, and all functional assays are typically global assays. One can tweak a bit with the inductors and the agonists, but on the whole, it is not easy to pinpoint from the aggregation pattern the direct platelet defect, unless Glanzmann or Bernard-Saultier, which is relatively easy. And here comes immunofluorescence a bit into the game. And here you see platelets from a blood smear, typical pseudopodia, staining for glycoprotein 2b3a. And this is a Glanzmann patient type 2, in which bit of the glycoprotein is still produced, but gathers inside the platelets. It is not exposed to the platelet surface because only the intact complex can be exposed. And here is an example of monoallelic Glanzmann thrombasthenia due to a large deletion in GP2B. And one can see in the direct comparison that staining of platelets is dimmer. But at this stage, I can also mention very clearly that subtle quantitative difference in glycoprotein expression is really difficult to judge on immunofluorescence. It's a semi-quantitative method at the best, and flow cytometry is definitely much better for the quantitative analysis. Other examples are disorders in which the platelet structure is affected, small platelets like in the Wiskott-Aldrich syndrome and X-linked thrombocytopenia, dynamin-associated thrombocytopenia, or those who don't stain by light microscopy, like the gray platelets in the gray platelet syndrome. And then we have the large group of the macrothrombocytopenias. I don't go into the details, and the list is definitely not complete, but I will show you a few examples how these different syndromes look like by immunofluorescence microscopy. Here again, an example of Glanzmann thrombasthenia. In the upper row of photos and pictures, you see always the control. GP2B3A is stained in green, and myosin as a counter stain is stained in magenta. And the color you should see for the certain structure is always indicated in the heading of the figure. In the typical Glanzmann patient, in whom 2B3A is more or less lacking, you see normal staining for myosin, but an absent or more or less absent signal for 2B3A. This is then an example for the Bernard-Saultier syndrome. You counterstain this time with von Willebrand factor, which you can nicely see in the alpha granules. Nice coverage of 1B9 on the membrane. And here, one of those patients with the typical European GP9 defect, which sometimes shows a bit of GP1B9 complex still on the platelet surface. And this allows at least also a semi-quantitative assessment whether still some of the glycoproteins are expressed. And you all know that patients who still show some of the glycoproteins on the surface have a much lower risk for producing ISO antibodies against the intact glycoprotein than those who have no proteins on the surface. This is in the lower part a GFI1B defect with strong staining for CD34, which is typically absent in the normal platelets. And I have to say, all these pictures are all taken from regular blood smears referred and sent by mail to the lab. This is a typical feature here for the Wiskott-Aldrich syndrome and other disorders in which the molecular defect results in a very rigid cytoskeleton, often forming no longer these typical ring-like structures, but twisted rings, sometimes the double shape of 8. Or if these rings in these very resistant tubulins are resistant towards cold, and a very simple to perform experiment is to store the blood before making the blood smears at room temperature and the other sample at 4 degrees, make 2 blood smears, and if the structure of the beta tubulin is still preserved after 4 degrees storing, then it is very indicative that the beta-1 tubulin cytoskeleton protein is very rigid. And on the other hand, the tubulin 1 defect, in which the cytoskeleton protein is not arranged in the typical ring structure, but is arranged like a ball of wool, also generating larger platelets. And these are all examples in which the diagnosis of an inherited platelet disorder can be made right away from the blood smear. These are then the storage pool deficiencies. Here you see a deficiency in thrombospondin in the alpha granules, here of a Willebrand factor marker of the alpha granules, and here not a complete absence, but a strong deficiency in P-selectin, and this is either a patient with a GATA1 defect or with a GFE1B defect. They often present with this phenotype, and the dense granule proteins like CD63 and LAMP2 can also be nicely visualized and seen by fluorescence microscopy. Personally, I feel that fluorescence microscopy staining of CD63 is even easier than staining them on a grid for electron microscopy. You can also use immunofluorescence for very specific phenotypes. This is the only patient up to now described in one of the Nature papers a few years ago from the British group, where we were involved in characterizing this patient together with Kathleen Fresson, a young boy who had a defect in the histone deacetylase 6, an X chromosome coded protein which deacetylizes the beta-1 tubulin, and this tubulin, the acetylated tubulin, also shows these twisted rings I have shown you before with the Wiskott-Aldrich syndrome. But if you stain with an antibody against acetylated beta-1 tubulin, there's a strong signal in the patient. These acetylated tubulins are more or less absent or very few of them expressed in normal controls. And the mother, who was heterozygous because she has a second X chromosome, showed an intermediate phenotype. Finally, this gene defect caused alteration of the promoter region of GATA1, and the platelets also had a decrease in the alpha granules and a GATA1-like phenotype, and the mother again had an intermediate phenotype. So here, the genetic diagnosis of an HDAC6 deficiency could be nicely confirmed, not functionally, but morphologically by immunofluorescence. And then we have sometimes very complex phenotypes. That's a Jacobson syndrome, and you can appreciate that the dense granules are decreased. On the right-hand side is always the control, that there's an alpha granule deficiency, and I apologize for the very poor quality. CD34 is weakly expressed on some of the platelets. And all these features have been described before by others, but can be used then to confirm the genetic diagnosis, or together with the genetic diagnosis can show the phenotype. And we sometimes even find very peculiar structures like this tether formation, or this expressing only GP2B through A and not GP1B9. We sometimes see this, but the clinical significance of this is currently unknown, and we are working on this to better understand what is going on here. But this also seems to be a very specific defect in some platelet patients with platelet disorders. Now, immunofluorescence has the big advantage to show nice pictures and sometimes impressive pictures. The question is, how subjective is it, and can one validate the validity of this method? And here we had a fantastic collaboration with the group in Denmark of Eva Leiner and the group of Pepe Rivera in Murcia in Spain, who collected their patients in their countries with suspected hereditary platelet disorders and performed the genetic diagnosis. And they sent us the blood smears of these patients, unstained blood smears, without any further information. So it was a clear, completely blinded assessment. And we were able to enroll 59 subjects from 43 families with a definite genetic diagnosis, 20 inherited platelet different platelet disorders. 18 of them also had thrombocytopenia, mean age 37 years. 75% of the subjects were thrombocytopenic, and significant bleeding symptoms were present in a bit less than half of the patients. So what we then did in Greifswald, we did run them through our typical pattern of different monoclonal antibodies, and those who are interested in the exact specification of the monoclonal antibodies are referred to this publication. There we list them all, including the code name, how to order them from the different manufacturers. We had 9 patients with a known immunofluorescence pattern, known because others have reported this before, MYH9, typical Bernard-Saultier syndrome, monoallelic Bernard-Saultier syndrome, GFE1B, Glanzmann thrombasthenia, filament-related thrombocytopenia, delta storage pool deficiency, some of them with an associated Hermansky-Pudlak syndrome, the tubulin 1 defect, and Wiskott-Aldrich syndrome. And here you can see that during a blinded assessment of these blood smears, we achieved the correct diagnosis in all but 3 cases. So this means in 92% of the individuals, which were these. 2 of them were monoallelic Burnett-Saultier syndrome. And here you see the subtle differences in expression of GP1B9 in comparison to the normal control. And as I have said before, quantitative assessment of, let's say, minor changes in protein expression is really difficult to achieve by immunofluorescence. And the others were patients with Wiskott-Aldrich syndrome. We simply overlooked the twisted rings during the first assessment, and when we then became unblinded and reassessed the slides, sure enough, we found them. This was also shows that a symptom can be there, but in a completely blinded way and fashion, it might be difficult to really find the specific changes. But once one has some more information, it is a complementary method to secure the diagnosis. Thus, immunofluorescence microscopy of blood smears with a certain set of monoclonal antibodies identifies the underlying cause in about 70% of patients who are currently diagnosed with a specific genetic defect by a genetic diagnosis. And a major advantage of this method is for certain that it can nicely characterize the platelet phenotype. And this has really advantages if the genetic results are unclear and give results which have not been proven before to be pathogenic and to be clearly disease-causing. However, in this exercise, there were also 11 inherited platelet disorders where no known immunofluorescence pattern or morphological changes of platelets are clearly described in the literature. And we found morphologic changes in many of them, but we still can't say whether these are specific or just simply occurred only in these few patients or families we have seen, while for the FLE1 mutation and the actinin-1 mutation, we did not find any changes by immunofluorescence at all, which is at least very well known for the actinin-1 mutation. Beside these structures, we have mutations of the transcription factors. You all know that these mutations are often associated either with dyserythropoiesis or even with an increased risk for leukemia, which raises some ethical issues, which will not be the topic of the talk today. I would like to share with you non-published findings. Carlo presented them at the ISTH meeting, and the manuscript is currently under revision, which was a chance finding. It all started when we analyzed non-muscular myosin 2A on the blood smears, which we have as a routine in the setup, especially to identify the inclusion bodies and the leukocytes in the MYH9 disorders. And we first thought all these red spots were impurities of our antibody, until we realized that this blood smear was from a patient with genetically proven GATA1 thrombocytopenia, and the patient also had a nice or very impressive anisopoikilocytosis in the red cells. And we found, as expected in GATA1 disorders, reduced alpha granule deficiencies, and now it's the other way around. The patient is in the upper panels and the controls are in the lower panels. But you can see that thrombospondin, P-selectin are less expressed, and even the von Willebrand factor, although the large platelets look a bit, let's say, more bright than the controls. It's simply due to the size of the platelets. And when we then further look for the location and counterstained with glycophorin A to mark the red cells, it became obvious that these inclusion bodies of non-muscular myosin are located in the red cells. It is well known that GATA1 also causes dyserythrocytopoiesis. We then underwent several exercises to quantify them by image evaluation, and there was always a highly significant and clearly differentiation between GATA1 patients and controls. And the question was then, how frequent is it? And this is now a panel of Bernard Saultier, ETV6, Fly1, Glanzmann thrombocytopenia, Filimin, DIAF1, the MECOM, the very rare MECOM mutation, INCARD26, IKZF5, the ETGA2 mutation, the nectinin-1, the MYH9 disorder, which was of special interest because there we have the clear inclusion bodies of non-muscular myosin 2A in the granulocytes, but not in the red cells. The Wiskott-Aldrich syndrome, the TAB1 thrombocytopenia, the SARC mutation causing to hyperactivation, RUNX1, and Hermansky-Pudlak syndrome, and none of them expressed the inclusion bodies, but GFE1B platelet red cells also have the same inclusion bodies, and it's well known that GFE1B is also associated with this erythropoiesis. We also found in several controls other very small of these inclusion bodies if there was highly reactive reticulocytosis. And I copy in here the GFE1B inclusion bodies. And it's even if it's difficult to see here, you can easily differentiate the two types of inclusion bodies. The GATA1 and GFE1B related thrombocytopenia inclusion bodies are larger and much brighter than those with the increased reticulocytosis. But if you have a patient who has a very high reticulocyte count, one needs to be a bit careful in interpreting the finding. Finally, our findings on ETV6 related thrombocytopenia just accepted in the British Journal of Hematology. ETV6 is a transcription factor located to the nucleus of the megakaryocytes, and the authors of the first paper in Nature Genetics describing this disorder clearly showed that in the megakaryocytes, in healthy megakaryocytes, the transcription factor is only in the nucleus, while in the affected patients, it's also in the cytoplasma of the megakaryocytes. So it was an easy assumption and hypothesis that if it is in the plasma of the megakaryocytes, we probably also find it in the plasma of platelets of the patients, because it's simply transferred into the platelets. And this is the case. One can visualize ETV6 in the platelets of affected patients. It depends a bit on the secondary staining antibody, whether the staining is relatively dim or strong, but it is clearly visible. And we had in the paper a whole series, I think, of 6 or 7 unrelated families who all showed the same phenotype. And then we started an international workshop on immunofluorescence to see how reproducible these findings are and whether one needs a microscope in Greifswald and experience in Greifswald to recognize this. There were 7 participating centers. These are now the first preliminary results I can show you. The numbers you see on the outcome slide do not refer to the order of the centers here, so it's completely random. And what you see on one glance in green, the diagnosis was perfectly made on the blood smear, and in red, the changes were not correctly interpreted for whatever reason. You can also see that we don't have received back all results of all groups, and we are really looking forward to get the final evaluation of this workshop. Bottom line, however, is there are some disorders which can be clearly seen. In others, there might be some difficulties, and we have to work out where are the differences and where did the difficulties occur to give some guidance how to potentially improve this. It's in the nature of someone working on this technique for many years that I have to show a certain enthusiasm, but immunofluorescence on blood smears also have limitations. It's very clear it is subjective. And the solution is automated analysis with the application of artificial intelligence. We started the first pilot project. It seems that it works, but it's ongoing, will be a lot of work, but this will be the way to go to objectify the analysis and to reduce the individual work burden to automate the analysis. As I've said several times, assessment of the expression is semi-quantitative, and the reduction of the expression intensity is sometimes very difficult to identify. If it's only a 25 to 30% reduction, flow cytometry is really better. What is a real challenge is the rarity of the disorders and the conclusion whether a finding in one or two different or just in one or two different families is really generalizable. Given this challenge, I'm really grateful to our collaborators in Denmark and Spain that they shared their patient blood smears over several years now with us, which allowed to gather a large enough number of patients to prove generalizability of at least some of the features. And currently, we cannot identify clear signal transduction molecule deficiencies yet, but we haven't tried it very carefully. But I doubt that with the ability of signal transduction molecules, sending in air-dried blood smears will really give definite results and answers. So, diagnosis of inherited platelet disorders are always a combination of careful clinical characterization, functional tests. I try to persuade you today that immunofluorescence phenotyping might belong to this. Next generation sequencing has an important role, and both techniques are not competitive, they are complementary. Immunofluorescence can specifically look at certain structures if a gene defect is found, and can also, if the geneticists can look for certain genetic changes, if immunofluorescence shows a certain phenotype. So both alone are much less powerful than the combination of both. And together, all these techniques provide us in 2023 with a clear molecular diagnosis, still only in about 50 to 55% of inherited thrombocytopenias and about 25 to 30% of the inherited platelet function disorders. So we all still have a lot to do to find tools to characterize the remaining patients. So the take-home messages are: blood smears are easy to obtain, require only a few microliters of blood, and can be shipped by regular mail, which is fantastic for pediatric patients, especially for the diagnosis in newborns with unclear thrombocytopenia. Immunofluorescence of blood smears allows characterizations of the platelet structure, which might be helpful. This allows the diagnosis in a considerable number of patients with inherited platelet disorders, especially with those with the most frequent inherited platelet disorders. In my opinion, immunofluorescence is extremely helpful in evaluating variants of unknown significance of those disorders where we know how the phenotype look like. However, it has considerable limitations. I described them, and immunofluorescence is not competitive with other approaches. This is simply complementary and can help to strengthen our armament to diagnose those patients. And there are a lot of people who collaborated with us and who contributed to this. I would like to highlight Carlo Sanninetti, who did most of the work and is the first author of most of the papers, but also our colleagues and friends from Denmark and especially from Spain, who are also today with us. And I thank you very much for your attention. I'm happy to take questions.
Thank you very much, Professor Greinacher. We already have two questions. Dr. Saultier, maybe you wish to ask the first question directly.
Yes, thank you. Thank you, Professor Greinacher, for the great talk. I wanted to know whether you tried to use the MYH10 staining in order to diagnose patients with whether RUNX1 or FLY1 or GATA1 deficiencies.
We tried, but we weren't very successful. But I would be extremely happy to receive a few slides of RUNX1 patients that we could try another approach, and also to use a few different monoclonal antibodies. The problem is we don't have a good positive control, and we don't know, if we don't see any staining, whether simply our antibody doesn't work in the setting of immunofluorescence. And even if an antibody works in flow cytometry, it's not clear that it also works on the blood smear. Now, if anyone has a RUNX1 patient and would be willing to share a few smears with us, then we could stain for MYH9 and start a new approach doing this. I think we have only one patient with RUNX1, and this is not enough to prove the diagnosis.
Thank you.
Thank you very much. Our next questions are from Joseph Rivera, who's asking, in the case of ETV6 presence in platelets, the cases tested carry ETV6 pathogenic variant, or you have also tested individuals with VUS on benign ETV6 variants?
Yeah, we did. And one was a VUS, and this had a pathogenic variant. And now I really have to refer to Carlo, whether he had enrolled known benign variants. Unfortunately, I'm a bit uncertain in the moment, but Carlo, probably you can comment on this. It's your paper. Could you probably allow Carlo to unmute? Miriam?
This should be done.
Okay. Yeah, probably.
Can you unmute?
You can't unmute. Well, why not take the next question until Carlo has solved the issue, or you type it into the chat, Carlo, then we can also get the information.
So Jose Riviera has another question. In the case of GFI1b. If I am right, CD34 is present for both benign and pathogenic variants. So IF checking of other markers may be also useful. What do you think?
I'm not certain whether I have ever seen a benign GFE1b mutation and assessed because they had no phenotype and no platelet problem. What we also assess for GFE1b disorders is for certain the alpha granules, and one can nicely see the partial alpha granule deficiency, not as pronounced as in the gray platelet syndrome, but they have the large platelets and they have the decrease in partial deficiency in convolutant factor and thrombospondin. And together with the GFE1b, this is usually a very clear, straightforward diagnosis. But I don't think that I have ever looked at individuals who have normal platelets, no large platelets, and a benign variant, because there was simply no reason for us to look at these platelets in an otherwise normal individual.
So Professor Alessi also had a question. Professor Alessi, do you wish to ask your question directly? You should be able to turn your microphone on.
Yes, I can ask.
Please go ahead. Please go ahead.
Yes. Okay. Yes. So yes, I thank you a lot for your presentation. It is a very interesting presentation because we have sometimes difficulties to evaluate the pathogenicity of some variants, and I think that it will be interesting to use immunofluorescence to confirm the pathogenicity of some variants. And for example, I have a question on tube 1 variant, because we detect sometimes some variant, sometimes missense variants. Sometimes they are pathogenic looking at the bioinformatic tools, but it's difficult to be sure that these variants are pathogenic, and this difficulties is of course encountered for different genes. It is sometimes easy if we have all the phenotype of the M1H9 disease, for example, but in other context, sometimes it's difficult. In filamin A also, difficult to be sure that the missense variation are involved in the disease. Usually truncated variants are involved, but not missense, and it would be very interesting to have immunofluorescence result to be sure of the pathogenicity of the variant. So I'm sure that you focus your work on the diagnosis way of these inherited disorders, but it would be very interesting to begin from the genetic to look at class 3 variant, and to see if you could improve the sensitivity of the diagnosis using immunofluorescence.
Yeah. So,
and
we would be extremely interested in doing this. And now we are a large group of platelet experts here this evening. Now, why don't we get together? You have the genetic testings. It is straightforward sending us blood smears. Pepe knows he sends us blood smears from Spain on a frequent basis. It works very well. And we can look at these platelets whether we see a specific phenotype. We can discuss whether we do this completely blinded, or whether you give us a hint which gene you are looking for. And we can then give the judgment pathogenic or non-pathogenic. And if you compare this then in the context of the clinical presentation. Great.
Yes, but I would be very interested in sending you some samples of some non-informative variant to get more information and more data of the involvement of this variant.
Yeah.
And is it also possible to know the top performers of antibodies? Because it is also difficult to because sometimes there are 3-4 different antibodies, and it's difficult to do all the experiment to compare the quality of these antibodies. So it will be very important also to share this information to help people to perform immunofluorescence if they want to do.
Absolutely, and we therefore give the detailed information of the antibodies with the exact code name and where we obtained them from in the supplementary material of each of the papers, that anyone can simply copy them. What we did not do is writing down which antibodies did not work sufficiently, but those which are there in the papers, these are the ones we are using.
Yeah. Okay.
Right.
Okay, it would be also very interesting that you give us the name of the people you need to contact if we want to send some slides. So if you could give us the name of the people, perhaps it's you. I don't know if
it's me, or
you
contact Carlo Saninetti. He's this is his big project, and he will take care for it.
Okay, so I will contact him. Thank you very much.
Yeah, you're most welcome.
If you have any other questions, remember to type them in the chat. So Paola Barrerini also asked whom to send the slides, but you've already answered this question.
Yeah. And given the rarity of these disorders, either we work together, then we can see whether any of these new methods is really worth or producing artifacts. If we try to solve these problems by ourselves, we will fail, because barely any one of us has more than 3 or 4 families of the same rare genotype. And this is still not enough to really exclude by chance an artifact. But if we find the same features in 10 different families, and especially if we then have variants of unknown significance of benign variants, and they don't show the phenotype, then it is pretty clear that we are on the right track.
Of course.
Well, on this note, we have another question. Perfect. So Paola Barrerini asks, have you done MPL-IF2?
You mean MPL receptor?
Yes.
Yeah. Okay. No, we haven't. But we have no patient in the moment where I have the suspicion of an MPL receptor deficiency. Simply not done yet.
Well, thank you very much. Thank you very much, Professor Greinacher.
Dr. Rivera has a question.
Oh, you should be able to turn your microphone on.
Right. Can you listen to me?
Sure.
I just want to make a comment and have your opinion on that. And another important issue that this procedure or this technique can help us is to know also how these different variants across the genes will act, because we might understand why, some variants located in a part of the gene might have some effects. Other variants in other part of the genes might have different effects, and we might, individualize the pathogenic or the deleterious effect of this variant. So just in terms of knowing the biology, the molecular mechanism of this variant, I think this procedure, being simple, can also help a lot. I don't know whether you agree with that.
Pepe, great suggestion, a great comment. And I should have addressed this also in the talk. For time reasons, I'll let it out because it has been reported before. We can predict quite precisely which region of the gene the MYH9 mutation is. If it's upstream, we have very small inclusion bodies, often located close to the nucleus, and as further downstream, the mutation is, as larger the inclusion bodies in the granulocytes become. And this is, as you know, associated with the risk of renal impairment in the further course of the disease. So that's let's say a type of a proof of principle that good morphological phenotyping can be associated with the biological impact of a mutation. And this is very relevant and extremely interesting also for the other genes and mutations. But there is absolutely simply no data on this. And if, again, as said before, if we team up, bring the different patients who are well characterized together, then we can see whether by this additional tool, we can add some more information, which can then trigger further investigations on the biochemical level. And we are all grateful if some of these patients donate blood to us, and as better we have specified our hypothesis, is better we will be then to design targeted experiments to really prove or disprove a certain hypothesis with a reasonable amount of blood.
Thank you very much. Is there any other questions from the audience?
Okay.
I don't think there is any more questions. Thank you very much, Professor Greinacher. Thank you very much to our coordinators tonight. And thank you to everyone who has attended this webinar. We look forward to the next session, and please join us. Thank you again very much. Have a nice evening, everyone.
Thank you very much for participating.
Thank you very much. Good evening.